Hello guys, I'm here to tell you a little bit about how to log into an instance of OpenStack and how to use the virtual machine and the environment that it provides. So I will mainly concentrate on next generation sequencing and the various tools that are available for NGS data analysis and processing. So these days you might be hearing a lot about machine learning and big data and things like that. So actually today people are using machine learning techniques to diagnose diseases and propose plausible cures and etc. So they are using what we call as biological big data for their analysis. But where exactly does this biological data come from? So through this series of tutorials, we will try to get a feel of what exactly biological data is and how to generate your own biological big data. The thing is, you do not need to install any special operating system or sophisticated software for this task, for we have already taken care of everything. Using OpenStack, we have set up an instance, a kind of a virtual machine which has all the necessary tools. We call it the NGS Toolkit, and we'll guide you on how to access this virtual machine and everything else that is needed. So let's head straight into the world of NGS or next generation sequencing. So first of all, those from the biology background must be aware of what sequencing is. For our friends from non-biological backgrounds, sequencing is actually the procedure to determine the order of nucleotides in which they appear in the genome. So there are four nucleotides A, T, G, and C, and it's the order in which they appear which determines everything about a particular individual. Everything right from the skin color for and in case of human beings whether they will be able to grow a beard or not, whether they will be male, female, everything. So these four nucleotides are common for all living things on this planet. It's the difference in the sequence and the number of the nucleotides which gives rise to the diversity that we see around us. So sequencing had long been a time-consuming and tedious process, actually. So and then comes in NGS, and we can now do sequencing like never before, and it's relatively cheap. NGS sort of parallelized the process of sequencing, so it has given us the ability to sequence from a million to a billion DNA molecules simultaneously in one cycle. So it has kind of revolutionized the entire field of research. So it has given rise to even various fields of research like personal medicines, genetic diseases, diagnosis, and clinical diagnostics. So we will not go into much inner details about NGS. For now, suffice it to say that NGS is here to stay. Thus, getting to know how to work with NGS data, how to build your own algorithms for analysis, and how to process NGS data is definitely a skill that you would like to add to your skill set. So now let's get our hands dirty and start off with logging into the required virtual machine. So let me just go to my terminal. Yeah, so here I'll just clear up the entire screen. Yeah, so I've cleared the entire screen. Now just type in the following command: ssh ubuntu at whatever the username is at the address. The address for our virtual machine is 172.168.101.196. Yep. It asks for password. Punch in the password, whatever it is. So yeah. We are into this in this NGS Toolkit environment. Let's see what all is in there. Okay, it shows us quite a few binaries, zipped files, text, Python, and stuff. So yeah, for now these do not concern us. What concerned us was the command here. So this ssh ubuntu. Okay, so what ssh does is it allows you to log into a remote system with the username and the address. We'll get more into this later on. Now this was how you can do in a Linux environment. But let's say if we do not have a Linux environment, what to do then? Pretty simple then as well. Just go open a Chrome tab, type in the address that you want to log into, which is 172.168.101.139/horizon. Yeah, here we are. So domain is default, username is my name, password is, again, I am not going to tell you something. Yeah, I will connect. So yeah, it shows us the virtual machine that has been created by the IITG technical board. The various instances that have already been created. It has Marauder, NGS Toolkit, Stud Web Server, Gaurav Console, Karthik. I don't know what these are. What concerns me is this NGS Toolkit. So we'll open this NGS Toolkit. We'll open the console. Yeah. So we have what we call as the console. And once you are on this page, all you have to do is click on the console and press enter, and there you go. Next, all you need to do is type in the username that you have been given, which in our case is Ubuntu. We'll type in the password, whatever it is, which will be known to you. And yeah, so we are inside this NGS Toolkit with the under. We'll list all the directories which are in there, and yeah, we have the same directories that as we had seen in our Linux terminal. So this would be all for today. Now let's come to the most important part of the video. If you like this video and the idea of having an online virtual environment and would like to give this a try, then kindly go to the link provided in the description box and fill up the form. We will create a user account for you, and if you have any questions, suggestions or feedback, feel free to comment. This would be all for the first video. Thank you all. Cheers and bye bye.